Hello, students. This is Dr. Faizan Mirza. I'll be covering questions related to transport and plants in this particular video. So this question, it came in between 2019. This is from variant 21. And the question shows to us that meristematic tissue found in growing regions of the plant, such as shoot tips. Meristematics, what are meristematic tissues? We do not study these here in A level, but they have given this term. So they actually give the description what meristematic cells are. So meristematic cells have a role similar to stem cells in animals. So we know stem cells, how they work in animals. So we need to just relate stem cells to meristematic cells. The difference is that stem cells are present in plants and meristematic cells are present in, stem cells are present in animals and meristematic cells are present in plants doing the same function as stem cells are doing in animals. So figure 4.1 shows the some stages of the formation of the mature phloem, sieve tube element, and companion cell. We know what the sieve tube element is and what's a companion cell from a meristematic cell. So there's a meristematic cell here, and eventually it's going to form cell, which will eventually form sieve tube and companion cell. We can just annotate this diagram for our own understanding. So what's happening here? The meristematic cell, you can see it's dividing. It's dividing into two cells which are similar to each, exactly alike, and this must be divided. This division must be by mitosis. This one cell then starts to differentiate. We know that's exactly what happens in stem cell. One of the cell, which the cell that divide, one of the cell will replace the stem cell, and it will keep on providing the stem cell for the next for providing the specialized cell for the next replacements, and the other cell, it will differentiate to form a specialized cell. So here you can see the cell differentiates. The cell differentiation is the process in which the cell will gain and lose some functions to basically start doing additional functions. So yeah, it will end up having some new structures, and it will end up having some lesser structures as it was having initially, and there will be shape changes associated with it as well. So you can see the cell starts to elongate. You can see the cell is elongating. It gets a vacuole of its own. This is the vacuole here, and this was the cell membrane and cell wall were already there. But yeah, the vacuole just came in. So that's the differentiation here. Then from F to G, this is what's happening. Then what's happening from G to H? The cell splits vertically. The cell splits vertically, and when this splits vertically, one of the cell is not having vacuole, so the vacuole goes away, and the other keeps having vacuole and the tonoplast. Now after H, by stage J, the cell H, it splits horizontally while the other cell stays as it was, although it loses its nucleus though. So you can see that there are again no vacuoles in these two cells that got split horizontally, and there is the nucleus is still retained, although in the other cell the nucleus goes away. And eventually this will form the sieve tube, and these will form the companion cells. Now the question exactly goes for their only cell V and F are the daughter cells produced by the meristematic cell divide. Explain why it's important for the daughter cells in cell E to be a meristematic cell. So why cell E was important, because cell E can then divide by mitosis to form two cells, one of which can then differentiate into a special cell, another can become a meristematic, that can stay as a meristematic cell, and it will be a cell that will keep on replacing the stem cells, the meristematic cells, as they keep on differentiating. Complete table 4.1 to describe the changes that are shown in figure 4.1. I think I've already described these changes. You can just mention these changes in the box here for getting the marks here. Explain how the structure of a mature sieve tube element is related to its function. So you need to link the structure to its function. So how is it related? Mature sieve tube elements is having limited of few organelles. We know that there is less cytoplasm, there is no nucleus. This provides more space available for transporting sucrose. Sieve tubes have sieve plates, and the function of they prevent the tube from collapsing or bulging. Sieve pores can get blocked. We know callose form, although this is not very important point for A levels consideration, but yes, it's worth mentioning. This prevents loss of sucrose from phloem. Plasmodesma are present in between sieve tube elements, and their companion cells allow sucrose to get loaded. So yeah, I think we have added enough points here. So that will help us gain enough marks. Describe the function of the companion cells in the transport of phloem. So you need to explain the function of companion cells in transporting phloem. Transporting phloem. So companion cell, they actively pump H+ ion into the cell wall of the neighboring source cell, which is the mesophyll cell, using energy from ATP breakdown. So yes, that's how active transport is used, and H+ is pumped out. On that side of the cell, H+ and sucrose, they enter the companion cell through co-transport protein, and sucrose then move into the sieve tube through the plasmodesma, and that's what we will be describing here. Let's move to the next question. Now this question came in May 2019, variant 22. I have annotated this diagram heavily to just explain it. Although if I'll just remove all these annotations, so you will be able to see what exactly diagram they gave and what were they expecting us when they gave this diagram to us. So what was actually given to us. So this diagram was given to us, and with this diagram given, they said that this is a low power image of a part of a common sunflower, and the part means it could be any part, and the structure B and A is labeled here. State with the reason whether figure 1.1 shows the section through a root or a stem. So why is it a root or a stem? So that's why I added annotations here. So if since I've removed the annotations here, you can see that the epidermis is here. You can see that this central region is having the pith, and you don't really see the xylem and phloem as they are arranged in the root. You do not see any endodermis. What do you see? You see these vascular bundles. They are arranged in the periphery. They are arranged regularly in cortex of the cell, and of this part shows the flow. The arrangement of xylem and phloem is in vascular bundles. So these are the things that you can do. I have just added these annotations because this is how the root transfer section should have looked like. And since this diagram doesn't look like this, so this is not a root. So this is not a root. This is a stem then. And how the stem looks like, you can recall the stem looks like something like this. The stem is always having the vascular bundles spread out around the periphery, and the vascular bundles are having sclerenchyma at the outer end. Then after sclerenchyma, you have phloem, then cambium, then xylem. That's one vascular bundle, and then you have cortex, and cortex is having collenchyma and parenchyma both. The outer region is having the collenchyma, and the inner region is having parenchyma, and this central part is the pith, so of the stem. Now, coming to this diagram, always keep in mind whenever we are looking at a vascular bundle, the vascular bundle will always have four features. And the four features will be, if you are going from the epidermis side, this is the epidermis at the top, this is the epidermis. So if you go from the epidermis towards the pith, if we are going from epidermis towards the pith, this is the direction I'm going. The first section of the vascular bundle that I'll come across will be a sclerenchyma fiber. So these are sclerenchyma fibers at the top. After sclerenchyma fibers, you'll be phloem. After phloem, you will have cambium, and after cambium, you will have xylem. So they are always arranged in that order. So just recalling that. So the fifth state is the reason why it's a section of whether it's a root or stem. So the section is from a stem, not root, because the vascular bundles can be seen or arranged in the central region, can be seen arranged, and central region is the pith having parenchyma cells. Cell type A is having a large central vacuole. We need to recall what a vacuole can do. So just with reason the role of this tissue formed by this type of cell. So you have to relate the tissue with the type of cell. So only then you will get the mark here. So these cells at the stem, they will store starch, because you need to recall what vacuole can do. It can store starch, it can hold water, it will have a hydrostatic pressure. These cells then get turgid, they can provide support to the stem. Name the structure B. The structure B is vascular bundle. The actual length of B is 650 micrometers. 650 micrometers need to be turned into a millimeter. So what do? You just divide it by 1,000. So whenever you are going from micrometer and you are going to the millimeter, you always divide by 1,000. And when you are coming from millimeter and you are going to micrometer, you will just multiply by 1,000. So in this case, this part is asked. So what we'll just do, we'll show the division and we'll mention the answer. Yeah. And then when structures are observed at high magnification, more details, xylem vessel element and so on, elements can be seen, safe tube elements can be seen. Outline the difference in the structure of xylem vessel element. You need to compare xylem vessel element with the phloem sieve tube element. So compare both the elements together. So xylem vessel element, it lacks cytoplasm, cross walls and walls are not there, organelles are not there, they have pits, but no plasmodesmata. These walls are thickened due to lignin. So that's what I can mention about them. Then coming to phloem. Phloem sieve tubes have little cytoplasm in their periphery. Few organelles are present and walls are present, having sieve pores, no pits, but plasmodesma is present. So what I've done, I have divided my answer in such a way that I have used half of my space for outlining what phloem sieve tube is doing. And I have mentioned the differentiating feature of xylem with phloem, xylem as element in the first half of my answer. You can go for like and like for like comparison as well, but I prefer this mode for my answer, and I found that to be more comfortable, although both are acceptable. Okay, so this question, it came in made in 2018 and it's made in 23, and this question deals with this xerophytic plant, and they are showing a photomicrograph, a section through a leaf of this plant here. The palisade mesophyll are this. So if these are palisade mesophyll, these are palisade mesophyll, these must be spongy mesophylls here. This must be a vascular bundle, have the vein. So this is a vascular bundle shown along its, you can say on its central axis. And this is the same vascular bundle shown to you along its length. So this is also a vascular bundle. So anyhow, coming back. So this is the lower epidermis, this is the upper epidermis. You can see the upper epidermis having multiple layers. The lower epidermis also having multiple layers. So that basically helps in preventing the loss of water. And there are this region, something like this. They have called that this is stomatal crypt. All this is mentioned in the diagram, shown in the diagram and given in the description. The leaf shown in figure 3.1 is a number of adaptations to reduce water loss by transpiration. Two of these adaptations are a multi-layered epidermis. So you can see, yes, there is a multi-layered epidermis. Stomata are found only in depressions deep inside. Depressions mean they are deep inside, known as stomatal crypts on the lower surface. So the upper surface lack stomata altogether, and the lower surface also lack stomata. The stomata is present only inside the stomatal crypts and nowhere else on the lower epidermis. Now what's happening? Explain how the multi-layered epidermis stomatal crypts will help in reducing water loss. So we know that the multi-layered this multi-layered region at the top, it increases the distance of water loss. It also reduces the water being lost through the cuticle from upper epidermis, so that will be water loss will be prevented. What's the function of the stomatal crypts here? The stomatal crypts, they are not exposed to the external air. The external air is here. This is where the external air. So external air will be very dry, and this dry air will not be in direct contact with the air inside the leaf, which is having high moisture. So the gradient is not a very sharp gradient. The sharp gradient is not coming in direct contact, and the water vapor here is coming in contact with the air which is present inside the crypt, and this region inside the crypt will be highly moist because it is having trichomes as well. These triangular structures, these hair like extensions, these are trichomes, and these trichomes, they will reabsorb whatever we were back and send it back into the leaf. So that's what we are mentioning here. So stomatals are not exposed to the external environment. Surrounding environment, water loss prevented. Inside the crypts, moisture will be high and prevent water loss by lowering the diffusion rate. So that's what you can mention here. Sucrose amino acids and other associates synthesized in pacimidophyll. They pass on to the vein. They are transported specialized cells from sucrose from source to sink. Name the cells specialized for transport of phloem assimilate. So yeah, there's few phloem C2 elements. That's a pretty straightforward factual answer. Explain the difference between the source and the sink. So source is a part of a plant capable of carrying out photosynthesis, producing sugars. Sink is where the parts of plant where the assimilates are either used up for the growing tips or they are stored in the roots of cortex. Okay, so this question came in Feb March 2022, variant 22, because there's this one variant in Feb March. So the question part of this question shows you a simplified diagram representing the transfer section of a young root. They have shown to you the diagram is not on the scale. Figure 1.1, draw label line to where the letter C to identify Casparian strip. So this is the root hair cell. This is the epidermal cell. So every epidermis cell is not having root hair. So this is an epidermal cell without a root hair, but this is a root hair cell having an extension. The extension is grown in between the soil particle. So any moisture which is present in between the soil particles will go into the root hair cell and it will be transported to the xylem. Now, this is xylem. How do you identify xylem? Xylem is always having a geometric shape, and the geometric shape polygonal. It will be polygonal. It will be having pointed ends, and it will be having a thick cell wall having lignin. Now, what the diagram shows to us? The diagram shows to us that water is being transported along the cell walls. You can see this pathway shown to us is the pathway of cell walls. So that definitely is apoplastic pathway. So if it's an apoplastic pathway, that shows that the pathway is through cell walls. Then this cell, this tissue, what will be the name of this tissue? The name of the tissue will be endodermis. And why would this be endodermis? Because we can recall that endodermis is having Casparian strip, and Casparian strip is having the deposits of suberin, and this suberin prevents the apoplast pathway. So water entry into the xylem is prevented through the Casparian strip. The apoplastic pathway will be blocked here. So the only pathway that will allow water to keep on moving will be symplastic pathway. So this is the Casparian strip. So you need to identify with the letter C. So we'll just point to the level that this rectangular structure here, that this is the letter C. This is our Casparian strip. Explain how the root hair measure is approximately 5 micrometer. So the distance, the root hair, and 500 micrometer in length. So this is about 5 micrometer in its width and 500 micrometer in its length. So this I've just annotated this on my diagram. So what this happening? Explain what does it tell? Explain how this adapts root hairs for the absorption of water. So this basically provides a larger surface area to volume ratio, and this large surface area to volume ratio of the cell is having extension that grows in between the soil particles to absorb more water. Name the pathway. So that's apoplast pathway. Okay, Water enters xylem vessel, that's shown in figure 1.1. Explain how water moves a xylem vessel to the leaves as a continuous column. So when you are mentioning as a continuous column, so we'll just have to give 3 marking points, and we, if it's a question of the question actually did come earlier for 5 marking points here. Now since it's for 3 marking points, so we just we'll just recall what's happening. So it's the, let's just the mind map. So try to recall transpiration pull, and transpiration pull allow what transpiration pull is exerted because water vapor is lost from the aerial parts of the plant. Xylem provide water to replace the water lost to the mesophyll. As a result, hydrostatic pressure decrease and water start rising up the xylem. When the water is rising up the xylem, this is because of addition between water molecules, and because of the addition of water molecule between walls of the xylem and water molecules, and cohesion between water molecules. And all these are because of the hydrogen bonding. Water start to rise as a continuous column. So that's what you can just you can explain here. That's it from my side. I hope that was helpful for you.